Hi, my name's Steph Szarmach, and I'm a graduate student at Penn State. In this video, I'll be comparing the effectiveness of different sequencing methods for characterizing divergence across the genome in non-model organisms. Whether you have experience working with genomic data or are just looking to start, I hope this video will be helpful for anyone interested in studying genomic divergence in species that are currently lacking genomic data. In this talk, I'm going to be sharing the results of a study where we compared genomic landscapes of divergence generated using 3 generations of data from the same study system, the yellow-rumped warblers. When we say genomic landscapes of divergence, what we mean is how genetic differentiation varies across the genome when we're comparing 2 species or populations. Identifying genomic regions that are highly divergent between closely related species can help us to understand what loci may play an important role in the speciation process. In one of the first studies of divergence landscapes in birds, Ellgren et al. Quantified fixed differences across the genomes of pied and collared flycatchers, which is shown here in the first figure. Here we can see chromosomes 4, 4a, and 5, and the peaks show regions with higher genomic divergence between the 2 species. Many studies have found that these divergence peaks align with genes related to traits important for species recognition, like plumage pigmentation. So we can see that quantifying genome-wide divergence can help to answer many evolutionary questions. But when designing a study, you have an important choice to make: which sequencing approach you'll use. The main difference between the methods we're going to discuss here is how much of the genome you're actually sequencing. Here, I will briefly outline the 3 methods that we compared in the study. All 3 of the methods start with the same input, sample DNA that we collected from a bird. The first method is double digest RAD-seq, which has the lowest marker density. In this method, we cut the DNA with 2 restriction enzymes and only sequence fragments that are cut by both of the enzymes. The second method is genotyping by sequencing, or GBS. Here, the genome is cut with 1 restriction enzyme, and all fragments cut by the enzyme are sequenced to a given length. The third method is whole genome resequencing, or WGS. The genome is randomly sheared, which we represent here with a paper shredder, and all of the fragments are sequenced. For all 3 methods, the fragment sequences are then aligned to a reference genome, and variable sites are identified for the downstream analysis. When choosing the sequencing approach for a project, trade-offs come into play between 3 main factors: the marker density, or how much of the genome is being sequenced; the depth, or how many sequencing reads cover each region of the genome; and the number of individuals that you can include on one sequencing lane. Given a fixed budget, increasing any one of these factors will require a reduction in the others. It's important to choose a method that will provide enough data to answer your research question while staying as cost-effective as possible. This problem brings us to the 2 main questions of our study. First, we wanted to determine which sequencing method is the most effective for characterizing genomic landscapes of divergence. Second, we wanted to compare the cost-effectiveness of the sequencing methods, specifically in terms of the cost per data site generated. To answer these questions, we characterized divergence landscapes between closely related subspecies in the yellow-rumped warbler species complex. Our study focused on the myrtle warbler, which is found in northern North America, and the Audubon's warbler, which is found in the West. The reason we chose to focus on yellow-rumped warblers is that we have 3 generations of sequencing data generated for this system, so we can directly compare how observed genomic landscapes of divergence differ depending on what type of data you use. So first, we have the double digest RAD-seq data, which has the lowest marker density, covering the smallest proportion of the genome. This data comes from 5 myrtle warblers and 5 Audubon's warblers. Then we have our genotyping by sequencing data, which has a medium marker density. Again, this data set samples 5 myrtle warblers but we have Audubon's warblers representing 2 different mitochondrial clades, which I'll come back to later in the talk. Finally, we have whole genome sequence data for 5 myrtle warblers and 5 Audubon's warblers from each mitochondrial clade at a depth of 4x across the genome. We ran all 3 of these data sets through the same bioinformatic pipeline to generate the landscapes of divergence. We use the program ANG-SD, which is designed for working with low coverage data to estimate FST across the genome using each data set. These Manhattan plots show the patterns of genomic divergence between myrtle warblers and Audubon's warblers that were estimated using each sequencing method. Our measure of divergence, FST, is on the y-axis, and the chromosome position is on the x-axis. Each point represents the FST calculated across a 10 kilobase region of the genome. You can see that the genotyping by sequencing and whole genome sequencing datasets allow us to see distinct peaks of elevated divergence. These peaks are not very clear using the ddRAD method. We can see there are windows with high FST, but the overall pattern of peaks is definitely not as clear. We can zoom into specific chromosomes for a more fine-scale look at how well each method captures the patterns of divergence. Here we have chromosome 1A. From the whole genome sequencing data, we can see a region of elevated divergence starting around 50 megabases that's actually composed of 2 peaks. We can also see this high FST region in the GBS data, but we aren't able to actually tell that there are 2 peaks very close together. The ddRAD data has a lot more noise. You can vaguely see where the peak is, but the pattern is not very clear. For another comparison, here's chromosome 5. The whole genome sequencing data reveals there are 5 peaks of elevated divergence, and we can clearly see where each peak begins and ends. From the GBS data, we can sort of see the first peak and some vague outlines of the other peaks, but it's much more difficult to tell where the peaks start and end. From the ddRAD data, we can see some areas that have higher divergence, but we would not be able to determine a distinct number of peaks. So we find from these comparisons that the GBS and whole genome sequencing methods can both give a good outline of the genomic landscapes of divergence, but whole genome sequencing is superior for revealing the fine-scale details of the peaks. So now we're interested in seeing how these methods compare in terms of cost effectiveness. So looking at the total cost for one sequencing lane, whole genome sequencing is the most expensive method, followed by GBS and then ddRAD. However, each method produced a different amount of data that we could use in the analysis. So we wanted to take that into account in our cost comparison. Here we show how many nucleotide sites were incorporated into the genome-wide FST analysis for each data set. When we calculated the cost per megabase of informative data sites, we found that whole genome sequencing and GBS are actually the most cost-effective in terms of the amount you're paying per site. One reason for this is that with ddRAD, we're sequencing a smaller proportion of the genome at a much higher depth, which leads to redundancy when the same base is sequenced many times without providing additional data. Unlike using reduced representation methods like ddRAD can also result in a higher cost if the marker density is just too low to accurately capture landscapes of divergence, as seen in the previous slides. There have been a number of studies that failed to detect any highly divergent SNPs using ddRAD, but then these peaks were recovered when whole genome data was used. So at that point, you've then paid for both ddRAD and whole genome sequencing. So even though ddRAD may be the cheapest option on a per lane basis, we think it's worthwhile to invest in whole genome data for characterizing landscapes of divergence because you're generating more data at a lower per base cost. In the remaining time, I'll discuss a specific case where whole genome sequencing was able to reveal a divergent region that wasn't detected using reduced representation sequencing. When I described our sampling scheme earlier in the talk, I mentioned that we sampled Audubon's warblers from 2 mitochondrial clades. Previous studies of mitochondrial DNA and yellow-rumped warblers found that there's a contact zone in the southwestern US between 2 deeply divergent mitochondrial lineages. Audubon's warblers in the southern part of the zone have ancestral haplotypes belonging to the same clade as the black-fronted warblers, while the Audubon's warblers in the north share haplotypes with myrtle warblers that were likely introduced through mitochondrial introgression. Past genotyping by sequencing didn't show any divergence in the nuclear DNA of Audubon's warblers in this contact zone. We were curious if this really meant that no nuclear introgression had occurred, or if the GBS data just didn't have the power to detect any divergent regions. We estimated FST across the genome between Audubon's warblers with the northern introgressed mitochondrial type and Audubon's warblers with the southern ancestral mitochondrial type. We found that divergence was low overall, but there were several regions with elevated FST, and the 2 highest were adjacent windows on chromosome 12. Here we show the peak of slightly elevated FST on chromosome 12, which was detected in the whole genome data, but not from genotyping by sequencing. When we explored what genes were located in that region, we found it contains NDU-FAF3, which is a nuclear encoded mitochondrial gene that encodes a protein in complex 1 of the electron transport chain. We're now exploring this region further to determine if it may have introgressed alongside the mitochondrial DNA. In this case, the whole genome data allowed us to identify a pattern of divergence that wasn't evident from our other data sets. So in conclusion, whole genome sequencing recovered the highest resolution genomic landscape of divergence. Whole genome sequencing was equally or more cost-effective than GBS or ddRAD in terms of the cost per informative data site. And only whole genome sequencing allowed us to identify small regions of elevated divergence between Audubon's warblers with divergent mitochondrial haplotypes. We find that while reduced representation methods are valuable for many other applications, we found that whole genome sequencing was the most advantageous approach for characterizing genome-wide divergence. With that, I'd like to give out some special thanks. First to the Museum of Southwestern Biology and the Cornell Museum of Vertebrates for tissue loans, to the Irwin Lab and the Levitt Lab where the original genomic datasets were generated, to the Penn State Eberly College of Sciences and Huck Institute of Life Sciences for funding the research. And then I'd like to thank my other members in the TADES lab. If you're interested in this topic and want to go on a deeper dive, I'd invite you to check out our paper in Molecular Ecology that was published earlier this year that gives the full story of this study. And if you have any questions or would just like to chat, I'd be happy to talk either through my email, which is listed below, or on Twitter. And thank you for watching this video.